Hey everybody, sorry my quick time crashed there, so I'm going to pick up where we left off. So we were just talking about karyotypes, and we had just gotten through describing the fact that we have um, different sets of chromosomes from the maternal and the paternal lineage, but then the X's are going to be exactly identical because they are actually going to be specific copies, identical copies of one another. And that we had all these nice pairs arranged by size until we got down to the sex chromosomes, and that's right where we cut off. So sex chromosomes are going to consist of an X and a Y in a male, and an X and an X in a female. And what's interesting is that these guys are actually going to align up during cell division as though they are the same, but they only carry information in the Y in terms of male sexual differentiation and development. But the female has a lot of important information on it. You can see it's actually a fairly large chromosome compared to some of the other chromosomes. And so that's actually going to become important later on, because what that means is that ladies, we have double the amount of information for this chromosome than gentlemen do, because the only thing on this Y that's important is basically the male differentiation and development cascade, and the rest of it is just going to be regions that are going to allow it to pair up with the X. But the X has, for example, color vision or blood clotting factors on there, and so we are going to have very special things in there that women are going to have two copies of. And women, what's interesting is that because we don't want to end up with too much of whatever protein can be created, we are going to intentionally shut down one copy of our X in each of our cells from a very early stage. That means that we're only going to end up expressing information from one of the X's. But if there happens to be something wrong with what's on that X, for example, one of the X's is going to have an abnormal blood clotting protein, then we'll shut down the one that is abnormal preferentially, which means that we're going to end up being able to select for whichever one has color vision or has the normal blood clotting factor. And that means that certain disease traits are going to be found more often in men who don't have the ability to compensate. They only have one X than in women. But I digress. All right, so that's what a karyotype is, and we're looking at, this case is the male individual. We're looking at all of the genes together, and we're looking at how they align with one another. Now, if you were to see, for example, a third copy of chromosome 21, you'd be looking at an individual with trisomy 21. So trisomy would be three copies, right? Normally we have two of each. Monosomy would be one copy of one of them, and that's not going to be something that's going to be well tolerated. In fact, most trisomies are not well tolerated too either. The only reason that this one is well tolerated is because the amount of information is so small. It's one of the smallest chromosomes. Okay, so generally, the homologous chromosomes are not identical. And what does that mean? That means that the maternal information is going to be different from the paternal information. Even though we're still showing genes for hair color and for eye color, they might be different alleles for that region. And how do we end up with these changes? These changes are called mutations, basically, and they happen when we are undergoing cell division and we end up with an error in replication that's not caught. And sometimes mutations are going to be what we call favorable, or sometimes they're not favorable. And the ones that are going to be able to endure, that means they're not going to kill the individuals that carry that trait, are going to be called alleles. And so something that's not going to affect, for example, the ability of an individual to survive and reproduce, like the color of your hair, right? Black, brown, or blonde hair, or the different types of songs that birds might sing. So you might have a different allele, but you would still have the same, you would still be in the same gene locus. And these changes are going to get passed on from generation to generation, right? This is why the expression apple doesn't fall far from the tree, and you do actually look very similar to some of your parents, right? Because whatever you got from your parents are going to be passed on to you and onto the next generation, etc. Now, sometimes mutations are going to be what we call deleterious or a negative consequence for the organism, something like sickle cell anemia, which is found in individuals with an African American background. That's going to mean that they're going to have a difficulty in carrying oxygen to their tissues because their blood cells actually have an oddly shaped sickle shape instead of being shaped kind of like a donut. Sometimes changes are beneficial to the organism, and that's going to allow them to survive and reproduce more successfully than other members of the species, which means that this is going to be a mutation that's going to be enduring. It's going to get passed on probably at a higher frequency in the subsequent generations. As we mentioned, the female and the male have different sex chromosomes, right? The female has two X chromosomes. They're going to appear very similar. The male has an X and a Y, but the X and the Y pair during meiotic cell division. So they have a region called the pseudo-autosomal region, which you don't need to know, but basically that allows the X and the Y to act like mates or pairing partners during meiotic cell division. All right, so if we have genes, right, this is gene 1. These are going to look the same because they have the same alleles. Gene 2, we have different alleles. So for example, this could be hair color, both brown. This could be eye color, one brown, one blue, right? So although we can have the exact same copy at that locus, we can also have two different copies at that locus. All right? Now sometimes we're going to have a specialized type of cells that are called haploid. Now, haploid cells are basically cells that come from sperm or that create sperm or eggs, right? And that's going to be cells that are going to make gametes. Gametes only have one member of each of the chromosome pairs. I mentioned it's not just a random division in half. It's one copy of chromosome 1, one copy of chromosome 2, one copy of chromosome 3, etc. When you normally would have two copies of each. So a haploid cell has half the number of chromosomes, but in a very ordered fashion of a diploid cell. And when those two come back together, then they're going to create a genetically unique organism, right? So that when the sperm and the egg come together, sperm fertilization, that's going to create fusion of the haploid cells, which creates a fertilized egg or a diploid cell. And that's going to have two copies of each of the chromosome, right? One copy from mom's lineage from the egg, one copy of one from the dad's lineage from the sperm. And the copy from the egg, of course, being maternal chromosome, from the sperm is the paternal chromosome. And another introduction I want to throw out here is something called N and 2N. So N would be the haploid chromosome number, and the diploid chromosome number is 2N. And that's because different animals have different amounts of our chromosomes, right? So for humans, we have 23 chromosome pairs, so a total of 46. But that's not exactly the same for, say, mice. They have 19, or Drosophila, the fruit fly, they have 4. But they're still going to have N being the haploid number and 2N being the diploid number. So when we talk about mitosis, we're not going to talk about the number of chromosomes. We're going to talk about half versus the full complement, right? So 2N being the complete pairs, and N being the haploid cells. Now, I mentioned that there was a specialized time called interphase where the cells are going to be replicating their DNA, undergoing growth, and acquiring enough nutrients to be able to get through mitosis. Right? Then we're going to enter into mitosis itself, which has several specialized steps as well. But the overview of cell division or mitosis is that we're going to get one copy of each chromosome, exactly one copy of each chromosome, right? So that means that we are not going to end up having 2 of chromosome 1 and an extra copy of, we're going to make sure that they go evenly, is my point. I apologize, I got distracted there. And we're also going to take half of the cytoplasm and approximately half of the organelles to be parceled out. This is not going to be as even as the separation of the chromosomes, which has to be very specific. And we'll show that when we talk about the metaphase spread. So interphase is the time frame when we're getting ready for cell division. And we have 3 major phases. The first is the growth phase. This is when we are acquiring our nutrients and we are getting ready to grow. We're actually going to be larger at this point, or getting ready to get larger at this point than normal cells. Then we undergo a synthesis phase, that's the S phase, during which time we're replicating every chromosome. There's also a checkpoint after the S phase where they make sure that the DNA is faithfully replicated. And if it is, then it enters into G2, which is a secondary growth phase, where the growth continues and preparation is going to be undergoing for the division of the cell, which is the mitosis, right? So during G1, we have 3 major things that have to happen. One, we're going to undergo a cellular growth period, and we're going to end up with specialty, especially occurring, so differentiation can occur, and then they have to make the decision whether or not to enter into S phase. If they are going to divide, they're going to have to enter into S phase. If a cell decides to divide, it's going to go into that S phase or synthesis phase, and that's going to be when it's replicating its DNA. From there, it's going to proceed to G2 after S phase. G2 is when it's going to continue growth and make the finalized proteins that are going to be necessary for the division of the cell. There are specialized control factors that are going to occur throughout the life of the organism, and this is important because if we don't have enough cell division, the body's going to fall apart because the development's going to falter, or the body parts aren't going to replace themselves properly when they get worn or damaged. But if we end up with too many cell divisions, we can end up undergoing cancer. So we have very specialized checkpoints that are going to stop cells from entering into cell division if they're not ready or if the DNA is not appropriately copied. All right, this is an overview of everything that we're going to be talking about today. Here's G1, S, and G2. All of that is considered interphase or preparation for mitosis. Mitosis itself is going to be comprised of prophase, which is basically setting the stage. Metaphase is when the action starts. Anaphase is when we're finishing things up. And then telophase and cytokinesis is going to be kind of like the cleanup after the show. There are several sets of factors that have to happen during each of these stages. So let me just hop right in here. So mitosis is going to be the actual cellular nuclear division. And it's going to be followed by cytokinesis, or the cytoplasmic division, which is basically when we're actually converting what was a large cell that now has 2 nuclei into 2 separate cells. Now, mitosis is very important. It happens in all eukaryotic organisms. It's happening in your body right now. It's how asexual reproduction occurs. So every time you cut yourself and you regenerate and repair, that's happening by the process of mitosis, right? And it happens pretty much immediately from the time that that the egg is fertilized, all the way up until the adult organism. It allows for maintenance, repair, and regeneration of your body parts, and it's also how our stem cells are going to reproduce. So it's very important. All right, so again, we have four major phases of mitosis, the first being prophase, the second being metaphase, third anaphase, and the fourth telophase. And when we get to lab, we're going to be able to look at the cell cycle. When we take a look at, for example, an allium root tip under the microscope, and we'll be able to see different root cells in various stages of mitosis. So the very first stage of mitosis is prophase. And as I mentioned, this is setting the stage. This is getting ready for the show. Several major events have to occur. First and foremost, we have to break down that nuclear envelope, right? We're never going to be able to get the spindle fibers to the chromosomes if the nuclear envelope is in the way. Secondarily, we have to make sure that all of our chromosomes are condensed down into those X's, right? They have to be nice and tight so that they're going to be able to be pulled to opposite sides. And they get pulled by these spindle microtubules. The spindle microtubules are going to be forming on either side of the cell. And then after the nuclear envelope breaks down, the chromosomes are condensed, the nucleolus is dissipated. Again, the nucleolus is the region where we're making ribosomes inside the nucleus. The nucleus is gone, nucleolus is also gone. And the spindle microtubules are going to start connecting to the central region of the chromosomes. So we are getting ready. The second thing that has to happen is that we have to have the microtubules attach to the centromeres. So spindle microtubules are going to come from the centrioles. Centrioles are found on opposite regions of the cell, all the way to the left and all the way to the right, or all the way at the top and all the way at the bottom, right? And the spindle microtubules are going to extend from the centrioles, which are at these specialized regions on the opposite sides of the cell, to the very center of the chromosomes at the centromere, and a specialized region called the kinetic core. So the kinetic core is a specialized region in the centromere, which again, that's at the center of that X shape. And the spindle microtubules are going to come from the poles to the nucleus and going to connect there at the kinetic core. Remember, the nucleus is dissolving at this point, so it's able to just kind of go right on through it, forms a basket basically around it. And then the duplicated chromosomes are going to get pulled apart. What does that look like? All right, so this is what the centrioles look like during interphase. Right? So during interphase, we're undergoing reproduction, and we've already gotten through the S phase because we're at late interphase now, so we've gotten through the growth phase. We are ready to enter into mitosis. So what happens? The nuclear envelope starts breaking down, right? The chromosomes start condensing from what was chromatin into what will be chromosomes. These are the spindle fibers, right? The spindle microtubules. They're coming, again, from the centrioles, which are going to migrate to opposite sides. You can see now they're at the top and the bottom in this image. And then they're going to connect at the centromere at a specialized region called the kinetic core, right? As all of this is happening, again, the nuclear envelope is breaking down, things are getting oriented, and we're going to enter into metaphase. Metaphase is when all of the chromosomes are aligned in what we call a beautiful metaphase plate. During metaphase, all the chromosomes are going to line up at the equator of the cell. And it's a very slippery slope, basically, from late prophase into metaphase, because prophase is basically when we're setting the stage, and as soon as the stage is set, then we're ready to begin. At this point, the kinetic cores of the chromosomes are going to be connected to the spindle microtubules. And again, the spindle microtubules connect to opposite poles of the cell. And all of them are going to line up in a pretty little spread. And when all of these are going to be in perfect alignment, then they're going to end up shortening. So what happens? The spindle fiber is going to either lengthen or shorten to be able to push everything or pull everything together until we end up in a perfect perpendicular line. And that's going to be the metaphase plate, right? Once that happens, and now we're all ready, everything is connected, then we enter into anaphase. And anaphase is basically when the sister chromatids, remember they were X's and now they're going to get pulled into L's, they're going to separate out and start being pulled to opposite poles of the cell. At the very first stage of anaphase, the sister chromatids are going to separate. So as soon as you see cells where the sister chromatids are pulling to opposite sides, it's into anaphase already. Then they become independent daughter chromosomes. So they're no longer sister chromatids. As soon as they separate from the X into two L's, they become chromosomes. And the kinetic cores are going to have specialized regions and the motor proteins that are going to pull the chromatids apart and push them towards the opposite poles, right? At the same time, we have specialized regions in the polar microtubules on the opposite poles that are going to end up pulling, right? And eventually that's going to end up making an oval shape. And after that happens, then we end up entering into telophase. So telophase basically is at the point when all of the sister chromatids, which are now their daughter chromosomes on opposite poles, have found their nucleoid region, which is going to become a nucleus, right? The nucleoli are going to reappear. The nucleus is going to start creating, so the nuclear membrane is going to start forming again. Chromosomes are going to start relaxing and going back into chromatin, and the spindle microtubules, which are no longer needed, are going to start disintegrating. Now, telophase and cytokinesis happen back to back. I could make it akin to walking out the door and the door shutting behind you. Basically, as soon as as telophase is happening, cytokinesis is happening as well. Cytokinesis is going to happen basically by tightening of the belt around the oval region to start pinching the membrane together until it separates into two. And because of the way that plant cells and cells are created, there's a major difference, right? Plant cells have a cell wall. And so it's going to have specialized steps to fill in that cell wall. So in animal cells, we're going to have microfilaments attached in a ring around the equator of a cell, similarly like to a belt that just keeps getting tightened and tightened. It's going to constrict the cell's equator and eventually create two daughter cells by pinching off. And that's going to enter into G1 of interphase. The whole cell cycle is going to be done then. But for the plant cells, it's going to be a little bit different because we're going to have to be putting in regions that are going to replace that cell wall. But I digress for a second. Let me finish the diagram here. So this is anaphase, and anaphase is what happens immediately after metaphase. Again, our sister chromatids are going to get pulled to opposite sides towards these towards the centriole regions, right, where the spindle fibers are connected here. And eventually, we're going to end up with telophase occurring, where we have constriction and the reappearance of our nuclear membrane and nucleoli reappearing, and also we're going to have the decondensation of the chromosome, so it's going to go back into chromatid. The same time that, again, cytokinesis, or the tightening of the belt, is going to occur, and then we're going to end up with two separate daughter cells. However, that was just showing you the animal cells. There's one major step that has to happen in a plant cell, and that's because we have plant walls, cell walls in the plant cells. And therefore, we're going to have to create a new cell wall in between the new cells. So we're going to do that by adding these little vesicles of carbohydrates between the daughter nuclei assembled along the equator to make a cell plate, and that looks kind of like this. So this is basically carbohydrates that are going to be coming out in little vesicles that are going to align along this region here, that are then going to create the cell plate, which is then going to create a cell wall, and the cell wall is also going to be lined by the plasma membranes because each of these vesicles is going to be surrounded by a membrane, so as they fuse, it's going to be laying the new membrane. So that's going to allow separation of the daughter cells in a plant cell. All right, so we have specialized proteins that are going to be responsible for the cell cycle, such as growth factors or cyclin-dependent kinases, which are called CDKs. And I'm not going to quiz you too heavily on this, but these are specialized enzymes that activate proteins either by phosphorylating, which means adding a phosphate group to it, or they can also remove different types of groups such as a phosphate, but kinases are specific to adding a phosphate, and that's going to inhibit the protein's activity. And CDKs are what's called cyclin dependent, and that's because they're actually only going to be active when they bind to specialized proteins called cyclins, and they're called cyclins because they only come about during specific stages of the cell cycle. Right? And this is what allows the cell cycle to be driven forward and not backward. So growth factors are going to bind to the surface of the cell, which leads to the creation of cyclin. Cyclin binds to CDKs, or cyclin-dependent kinases, which then get activated. And then this whole complex stimulates the activation of the cell into S phase so that it can start DNA synthesis. So when a cell is targeted for division, it's basically being given the information by growth factors and other information that's external, which then activate internal things like the specific CDKs, which are going to cause the cell to enter into the division phase. Now, CDKs are not just special to the S phase. We also have it during G2 and mitosis, and they activate specific events like the chromosome condensation or the nuclear membrane breakdown or the spindle formation. All of these things are specialty to specific stages of the mitotic cell cycle, and that's important because it drives it forward and never backward, because CDKs happen in a path, right? So the first ones get turned on, then the second ones get turned on, then the third ones get turned on, and in this way, we're stimulating each of the stages to occur during the cell cycle. And that looks kind of like this. These are growth factors that are going to be found in the external environment. They bind to a receptor and then create cyclins, which activate the cyclin-dependent kinase, which stimulates the replication of DNA. As I mentioned, there are specialized checkpoints that occur throughout the cell cycle, and that is also driven by these cyclin-dependent kinases. So there's several checkpoints that ensure multiple things. One, that the cell has completed the DNA replication successfully, that there aren't any major errors in mitosis. And there aren't any major errors in DNA replication, which would mean that they could not enter into mitosis. Additionally, that the chromosomes are undergoing the correct motions during mitotic cell division. So they're aligning properly and they're separating out properly. And if they end up not aligning properly or not separating out properly, these cells are going to be targeted for destruction. All right, so the three major checkpoints are at the juncture from G1 to S. So basically, if you don't pass this one, then the cell is going to survive. It's just not going to synthesize its DNA, right? And that's basically asking, is your DNA worth copying? And if it's found that it is, then we'll enter into S phase. And after S phase, we'll go into G2 and have another set of checkpoints from G2 into mitosis. And that's basically saying, is the DNA faithfully replicated? Are you the right cell to enter into this stage, etc.? And then lastly, there's a checkpoint between metaphase and anaphase, and that's basically making sure that every single chromosome is going to the right side. And if two are going one way and none to the other, then we're going to end up targeting that cell for destruction or apoptosis. So these three major checkpoints are going to help regulate the ability of the cell to enter into and proceed through the cell cycle. And these are actually checkpoints that cancer cells can circumnavigate. So they can get away with having abnormally replicated DNA, for example, and still enter into the cell cycle. So again, that first checkpoint ensures the DNA is suitable for replication. The second checkpoint from G2 to mitosis ensures the DNA has been replicated properly. And that last checkpoint from metaphase to anaphase ensures that all the chromosomes are attached properly, aligned properly, and going to the correct sides so that the anaphase can continue. So these are the major checkpoints in control of the cell cycle. Again, it's going to be from G1 to S, from G2 into the mitosis or prophase, right? So from G2 into M, and then from metaphase into anaphase. Now, we just talked about mitosis, but there are other ways which organisms can reproduce, for example, asexual reproduction, and it can be very successful, but the problem is that we get genetically identical offspring, and that means that if everyone has the same genes and are all genetically identical, that population can be susceptible to environmental disasters like a plague, for example. Whereas sexual reproduction allows genetic shuffling of the genes, which means that all of the organisms are going to be genetically unique from one another, which means that there's a little bit of variability in the population. So if a disease comes through, hopefully there's someone that's able to rise to the challenge so that the entire population doesn't die off. All right, so sexual reproduction is very important because it allows for the combination of parental alleles. It allows for combining genetic traits, right? And it also allows for mutations. Even though the mutation rate is very low, we are still able to create new alleles through the rate of mutation. And when these useful alleles are able to be combined through sexual reproduction, that means that evolution can occur at a faster rate, right? So we have individuals that have better chances of survival and reproduction than others. And this allows for the combination of alleles to change. And that means that the organisms, and it says adapt organisms, but just to be clear, organisms themselves don't adapt. The population as a whole adapts to a challenge of a changing environment. Okay, so we just talked about mitosis. So now let's talk about meiosis for a second. So meiotic cell division is basically sexual reproduction. And meiosis is a specialized cell division that creates haploid gametes. So what's haploid, right? We just talked about haploid and diploid. Diploid meaning that we have 2 N, haploid meaning that we have N. So for humans, diploid would be 46 and haploid would be 23. But it's not just a random division of those 46 chromosomes into 23 chromosomes, right? It's 1 copy of chromosome 1 and 1 copy of chromosome 2 and 1 copy of chromosome 3, etc. So basically, each gamete receives 1 member of each of the pair of homologous chromosomes, and then when they restore the genetic complement by meeting another gamete, they're going to have 2 copies of each of them. So let's talk about how this works. So basically, meiosis has 2 sets of division instead of 1. So everything that we just talked about, prophase, metaphase, anaphase, telophase, for mitosis, meiosis has prophase 1, metaphase 1, etc. So we also have prophase, metaphase, anaphase, telophase 2. So in the first nuclear division, we're separating out pairs of homologs, and each daughter cell receives 1 of the pair. Each daughter nucleus is considered to be haploid, even though it has 2 chromosomes, because remember, when that chromosome is in an X, it's still considered a chromosome. When it separates out into L's, then it's considered 2 chromosomes. So even though it has an X, it's actually going to be considered haploid. The second nuclear division, which is meiosis II, separates the chromatids into each of the daughter nuclei, two or more daughter nuclei, and it's going to end up creating haploid cells from a single diploid parent cell. So let me back up. Mitosis creates two diploid, genetically identical cells from a single parental cell. Meiosis creates four haploid, genetically unique cells from a single diploid parental cell. All right, so how does that work? So first we're going to undergo one set of meiosis where we're going to have our sister chromatids separate out into two pre-daughter cells, I guess. These guys are then going to separate out further under meiosis II, where the X is going to be split into two L's. So each of what was this all was considered one chromosome, and then it gets split into two. And then we're going to end up again with haploid. So four haploid cells at the end. Then what happens? Well, we end up with the diploid parental cells, mom and dad, right? They each make, mom makes an egg, dad makes a sperm. They come together during fertilization. So our haploid gametes, which each have N or half of the genetic material, come together to make the 2N or the full genetic complement in the diploid fertilized egg. And from here, the process of mitosis takes over. So from here, that cell is going to divide and divide and divide and divide until it finally becomes the embryo and then the developing organism. Now, the phases of meiosis, as I mentioned, they have the same names as the ones of mitosis, except they have 1 or 2 after them. So if you ever see a 1 or a 2 after prophase, metaphase, anaphase, telophase, you know you're dealing with meiosis. And we only have one duplication event. So if you picture that you have one duplication and one division, you get back to where you started, and that's mitosis. But in meiosis, we have one duplication and two division events. So we end up doubling and then having and having, which means that we end up with half of what we originally started with. So meiosis basically starts with one round of DNA replication and then followed by 2 rounds of nuclear division. The first round of division is going to separate out the homologous chromosomes, and then the sister chromatids are going to get separated out during the second round of division. So when you have a single round of DNA replication, you're going to make 4 chromatids of each of the chromosomes. Then we're going to divide them in 2 and then down into 1. During prophase 1, the homologous chromosome pairs line up. Remember, in mitosis, homologous chromosomes move independently, so that independent motion is not occurring here, and this is important because we're going to end up with something called crossing over. So maternal and paternal homologs are going to line up along the entire length. Again, maternal from the egg, paternal from the sperm, as you might imagine. And something called crossing over or recombination happens. And basically what happens is the paternal DNA gets spliced into the maternal DNA and vice versa. And so enzymes are going to cut here and cut there and then cross them back together and graft the ends together. And this is going to switch the maternal and the paternal DNA. Now, they happen at the exact same region, and this is important because we can't go crossing the genes for hair color with the genes for eye color, right? We're only crossing hair color for hair color and eye color for eye color. And so we have maternal and paternal exchange of information at these particular regions, and that's called recombination. And it's only called recombination if we actually are going to have different alleles that are transferred. So if they have, for example, a population where everyone is like lab rats and they all have the same genetic background, then allelic recombination isn't actually occurring because they are sharing the same alleles. But if the exchange segments have different alleles, we have something called genetic recombination occurring or crossing over. And we have several sets, 2 or 3 sets of chiasma for each of your pairs of chromosomal homologs. That means that if we have 20, we have approximately 60 different sets of crossing over that occurs during prophase 1. So there's a lot of shuffling that occurs. And we have an entanglement, basically, of these chromosomes at these chiasmatic regions. And they're going to stay together until they're pulled apart during anaphase. And just like any of the stages of mitosis, during prophase, we're going to end up breaking down the nuclear envelope, creating the spindle microtubules. Microtubules are going to bind to the chromosome. So a lot of the same preparation is going to be occurring, except we're going to have a major difference in that we are having crossing over occurring at prophase 1. Again, meiosis is going to reduce the chromosome number into haploid gametes, like eggs and sperm, so they're only going to be N. Then they get fused together during the fertilization event. That's going to make a diploid zygote, or 2N. From that point, that zygote is undergoing mitosis to create a multicellular organism. Now, this is important because if we didn't have our chromosomal number prior to reproduction, then sexual reproduction would result in doubling the chromosomal number each generation, which would very quickly lead to inviability. So we have to make sure that we have the number to restore the normal genetic complement. All right, so a lot of the same players are happening during prophase 1 and metaphase 1, right? So during prophase 1, we're breaking down our nuclear membrane. We are also going to have our spindle microtubules forming, and we're going to be aligning up during metaphase at the kinetochore. But we're having crossing over occurring here between the maternal and the paternal chromosomal homologs. And then we're going to have separation of the homologs during anaphase 1. And you can see this crossing over event is depicted by the fact that we have purple at the tip here and yellow at the tip there. So they've crossed and shared information. And then when we undergo telophase 1, we're going to end up with a cytokinesis event, and we're going to end up with these guys still as their own homologs. Then in the second set of it, we're going to separate out those into sister chromatids. So this is another up close of recombination, or also crossing over. And basically what happens is our sister chromatids are going to, so this is sister chromatids of one homolog, this is the second homolog. Again, we're seeing that these are both purple and these are both yellow. And then when we align, the purple crosses over with the yellow here and there. And this is only showing 2, but it happens approximately 2 to 3 times per chromosomal pair. And that's going to result in the individual chromosomes later on being a little bit different. So this one's going to be all purple. This one's going to be mainly purple with yellow tips. This is going to be all yellow. This is going to be mainly yellow with purple tips. So all 4 of them are going to be genetically unique. All right. After this occurs, we enter into metaphase 1. And it's going to be very similar to what we saw previously with regular metaphase. The spindle microtubules attach at the kinetochore regions, and the chromosomes are going to get pulled. In this case, the homologous pairs are going to get pulled to opposite sides. So the chromosomes are going to line up as pairs instead of as individual homologs, but that's it. That's one of the major differences. And then all of the same things happen. Chromosome homologs get lined up and get pulled to opposite sides during anaphase. However, the fact that we have a random combination of both maternal and paternal homologs without even having any of that crossing over that we just talked about, that's called independent assortment. And that's like flipping a coin to determine whether or not you get a maternal or a paternal homolog for chromosome 1. Basically, when the egg is being created, we're determining whether or not in this case it would be the grandparent, right? So the grandfather's or the grandmother's copy of chromosome 1 goes into one copy of the egg, and the other one goes into the other copy of an egg. And that's independently assorting. And so we can basically get either one or the other copy of chromosome 1, one or the other copy of chromosome 2. And if you do this together, it's basically 2 to the nth. And that's going to end up with about 8 million different combinations that can occur just from one set of parents. All right, so I digress. My point is that we have multiple different ways that we can end up with genetic variability. One is from what's called crossing over, which we just saw in prophase 1, and the other is from independent assortment, which is the independent alignment of chromosome 1 into one of the sperm cells and into the other copy of chromosome 1 into a different sperm cell. But I digress. All right, so anaphase 1 occurs immediately after prophase 1, and this is going to happen basically when the homologous chromosome pairs separate out from one side to the other, and the entire duplicated homologous chromosome separates. Now remember, in anaphase of mitosis is a little bit different because the sister chromatids are going to separate out. But in anaphase 1 of meiosis, the chromatids remain attached and move as their own homologous unit. And again, they're going to be pulled by microtubules. So each of the cluster is going to contain one duplicated member of each of the pairs of homologous chromosomes, and then when we enter into prophase 2, we're going to end up separating everything out again into their sister chromatids. So during telophase 2, which is the very end stage of the very first part of meiosis, so meiosis 1, we're going to end up creating these haploid clusters of duplicated chromosomes, right? Spindle microtubules are going to disappear. We're also going to undergo cytokinesis, or the cellular division. Sometimes nuclear envelopes may form, but generally this is going to be skipped because we're entering immediately into a secondary set of division. Telophase 1 is generally going to be followed immediately by a meiosis 2. There's no intervening interphase, and generally we don't even allow certain things like the nuclear envelopes to reform because we're just going to be going immediately into meiosis 2, so it would be kind of a waste. There is no chromosomal replication between meiosis 1 and meiosis 2, just to be clear, and that's going to end up with haploid cells resulting at the end of meiosis. All right, cells are actually considered haploid at the end of meiosis 1 because homologous chromosome pairs are considered to be themselves only one chromosome until they separate out. During meiosis 2, the haploid cells are going to be undergoing a secondary set of division. So the sister chromatids that were previously homologous chromosomes, right, are going to be pulled apart, virtually identical to mitosis. We do not have any crossing over events occurring, etc. So it's basically going to be very similar to what we see in mitosis. We end up with recondensation of the chromosomes if they decondensed at all. Sometimes they don't, because why would they? We're entering straight into a secondary division phase. Sometimes the spindle microtubules have broken down. Sometimes they are just going to be there already, and they're going to be able to do the secondary objective, right, which is capturing the chromosomes and pulling them to opposite sides. That again is going to occur at the kinetic core region of the chromatids, which is found in the centromere. During metaphase 2, duplicated chromosomes line up. Again, they're going to line up in the metaphase plate and separate out during anaphase, where they're moving to opposite poles, and in telophase 2, now we are absolutely going to have the decondensation of the chromosomes and the recreation of the nuclear membrane, and that's going to be followed by cytokinesis 2. And at the end of this, we're going to end up with 4 haploid cells. So 4 genetically unique cells with half of the DNA from the diploid cell. This is an overview of meiosis too. It's everything I just told you. Basically, we have these sister chromatids, which are now going to line up with that metaphase plate during metaphase 2. During anaphase 2, the sister chromatids separate out and become daughter chromosomes, and are going to go to their opposite poles. Then we have a tightening of the belt during telophase 2 and cytokinesis immediately after that, resulting in 4 haploid cells. Right, so these are all genetically unique haploid cells as well. If you look at any of the chromosomes, right, looking at this yellow one has a little purple. This one is all yellow. This one has yellow, but it's purple at the other region. This one's all yellow. Same thing here. So we're going to end up with different copies of the chromosomes, different arrangement of the chromosomes within each one of these specialty cells. All right, major differences between mitosis and meiosis. All right, so this just basically is going to go over multiple information for mitosis and meiosis. In mitotic cell division, it's going to be all of your body cells except the specialty gamete producing cells. That would be your testes, for example, or your ovaries, where you were creating sperm and eggs. The final chromosome number of mitotic cell division, it produces diploid cells, right? It produces 2 diploid cells that are genetically identical to the parent and to each other. At the end of meiosis, we end up with 4 haploid cells that are completely genetically unique, right? We have 1 set of cell division in mitosis, we have 2 sets in meiosis, and the production of gametes is the point here. Whereas meiosis, again, is creating a sperm and egg. Mitosis is creating all of the rest of the cells in your body. Repair of the cells, asexual reproduction, maintenance of your tissues, development, etc. Okay? So just to summarize, meiotic cell division is going to occur in specialized organs, ovaries and testes, and it's going to create specialized cells, sperm and eggs, which are a prerequisite for sexual reproduction for eukaryotic organisms. And meiotic cell division undergoes 1 set of replication and 2 rounds of division. So 2 division steps produce 4 daughter cells that become gametes. They are genetically unique and only have half of the information, so they are haploid. All right, when 2 haploid cells from different parents come together during fertilization, that creates a diploid cell that has specialty genetics that are going to be unique to everyone, including their offspring and their parents. And then when meiotic cell division occurs again, we end up with haploid cells again. So when this organism grows up, and then it's going to create its own haploid cells, which then is going to then fuse again and create another multicellular body, right? Mitotic cell division occurs when we are going to have growth or repair or development, or also an asexual reproduction. Okay, so by the end of this, you should absolutely know the difference between mitosis and meiosis. You should know the stages of mitosis, the different stages of meiosis, the major differences between the two, such as how many cells they create. Are those cells haploid? Are they diploid? Are they genetically identical? Are they genetically the same? Etc. Okay, so there's a huge difference in multiple life cycles of different eukaryotes. And there's 3 major aspects of the cycles that can be changed for this. So we can have the interval between meiotic cell division and the fusion of haploid cells can be very long or very short. Or the points in the life cycle when you enter into mitotic cell division or meiotic cell division can be very different. Or the amount of time that you spend in the diploid stage versus the haploid stage can be very different. And we're specifically referring to plants, I think, for this one here. And my point is that this is something that is allowing for reproduction, but it can occur at multiple different stages within the organism. Now, animals, such as us, right, we spend most of our time in the diploid state. So most of our cells are in a diploid state, with the exception of the sperm and eggs that might be residing in our bodies. And haploid gametes, again, these are constantly being created by meiotic cell division, sperm and eggs. They're going to come together to fuse to form a diploid fertilized egg. That's immediately going to be called a zygote. From that point on, the zygote is going to grow by the process of mitosis and differentiation. So this is what the human life cycle looks like. So, I mean, you should be pretty familiar, right? Let's start here with the two adults. The two adults undergo meiosis. That means that they create eggs in the women and sperm in the men. The eggs and the sperm fuse together to create a zygote. This, by the way, is the process by which Dolly bypassed. Dolly was the sheep that I was talking about that had the telomeric aging effect. They just took out this nucleus here and they popped in a nucleus of an adult animal, and then they induced this to occur, and they bypassed a specific stage that occurs just prior to meiotic division, whereby they elongate those telomeres. They didn't know that was a thing. That's how they figured it out. But I digress. So we go from the zygote stage. So again, this is when we are fusing an egg and a sperm. So we're fusing the haploid gametes into a diploid zygote. And then that zygote is going to undergo mitosis and development and differentiation and growth until it enters into the adult phase, whereby it undergoes meiosis again. So that's a human life cycle. But in plants, we have something that's a little bit different. It's called alternation of generations, basically. And we can have both the multicellular diploid and a multicellular haploid body. And so humans don't have a multicellular haploid body. But basically, this is going to be the process of creating sperms, right? Spores. So a multicellular diploid body is going to give rise to something called spores, right? They're haploid through meiosis. And then those spores undergo mitosis and differentiation to produce an adult haploid generation. That haploid generation of an adult eventually creates haploid gametes, which come together to create diploid zygotes. And then the diploid zygote grows by mitotic cell division to the diploid generation. So it's alternating. One generation is diploid, one generation is haploid. And this is specific to plants such as ferns. So plants have, like ferns, they have both the haploid and the diploid stages that are both going to be free-living, independent types of plants. But some plants, and again, I know we're going a little bit more into botany here, like flowering plants, only have haploid stages that are in reduced time frames. For example, haploid stages is the pollen, or the stamen, right? So here we go. This is going to be alternation of generations. Here we have a fern. The fern is the diploid adult stage, 2N. It's going to undergo meiotic cell division and create spores. Spores are N, so they're haploid. These haploid cells are actually going to undergo their own cell division, differentiation, and growth and create a multicellular organism that is all haploid. So all of these cells are going to be half the amount of DNA. And then one of those cells is going to be a specialty cell that creates an egg or a sperm. Those come together and fuse, create a 2N zygote, which is then going to be a multicellular diploid organism after the process of mitotic cell division, differentiation, and growth. So we're going to alternate back and forth between a diploid organism and a haploid adult organism. Again, this is specialized to plants. But my point is just to demonstrate to you that we can have these happen at different stages of the life cycle. It doesn't necessarily all have to be the way that we think of it when we think of animal reproduction and development. Now, genetic variability is very important to survive in a changing environment. So we're basically going to have variability within a population so that someone in that population is able to rise to whatever the environmental challenge might be. And we can do this by way of mutations, and mutations happen very rarely, but they do produce new variations, and this allows for, again, variability from one generation to the next. And that's a result almost entirely of meiosis and sexual reproduction. So mitosis doesn't really allow for much variability, although we do end up with mutations just by regular cell division, but generally those are going to occur in your somatic cells, and so they're not going to be passed on to your offspring. Again, we talked about shuffling of your homologs is going to allow the combination of chromosomes to be random. So it's a random lineup at that metaphase plate, which means that your chromosomes can line up either maternal-paternal or paternal-maternal in terms of going left and right. And that means that we have basically a total random flipping of the coin for each of the chromosomes. And that's going to increase the amount of independent assortment. So you're going to have a ton of different options of how you could independently assort those chromosomes. And that is only going to increase as the individuals end up with a higher amount of chromosomal pairs. So you have way less options of a way the pairs can dissociate. If you're talking about, for example, Drosophila, which has only 4. But if you're talking about something like a human organism, which would have 23 pairs, then you can end up with 2 to whatever the n is. In this case, n being 23, it's going to get larger and larger. So you could imagine 2 to the 4th is much smaller than 2 to the 23rd. And 2 to the 23rd actually turns out to be 8 million different possible combinations of chromosomes that can just occur from one individual. So were 8 million different possible combinations of sperm that your father could have created and 8 million different combinations of eggs that your mother could have created. And when you put those two together, 8 million times 8 million, that is the probability of creating you as the genetically unique individual that you are. And this is just showing you the combinations that are possible if we are only using three separate chromosomes. So maternal shown in purple, paternal shown in yellow, and we can have three maternal going one way, three paternal going the other way. We can have two and one, two and one. We can have the opposite, one and two, or we can have three, right? So two and one this way. So there's multiple different possible combinations, and that's only using three. If we were to look at, say, 23 pairs, again, we'd end up with over 8 million different combinations. So that's independent assortment. And that combined with the crossing over that we see in prophase 1 allows a complete novel combination of genes. So the resulting gametes are going to be completely independent of all of the rest of the gametes that were created in terms of genetic variability. So this allows for the introduction of new allelic combinations that might be completely new to the organism or to the population as a whole. And this allows for the shuffling, basically, of maternal and paternal chromosomes to ensure that we have no paternal or maternal chromosomes that are completely pure in the lineage within human populations. Now, if you figure that if we had 8 million combinations of sperm and 8 million combinations of eggs, and then we're only going to pick one, right, to come together during fertilization, that means that we're going to end up with 64 trillion different combinations that can produce you, again, as the genetically unique individual that you are today. And that's going to ensure that each human individual is absolutely genetically unique, both from its siblings and from its parents. And of course, the only exception to this would be identical twins, right? So I appreciate you listening to my lecture today. Aloha and happy